go.
They all have to sit that side. That side.
Otherwise, you can't see you.
Hello.
Hi.
Yes, probably.
Huh? 'What? Well, I mean, everybody's on high alert, but nothing is happening. So I presume something either went out with this heat, or Margaret is just not strong enough to carry the heat for the last 3 days.
You can see us.
6 months and then 3 months, and then 3 months again. Luckily she can't see me.
I can't hear you though.
Is the YouTube working?
It seems so. Currently, we have 8 other viewers.
Okay.
So hello to them. I don't know who you are. I just see that you're viewers. But hi, thanks for joining us.
You just have to be a bit patient between the flick of the slides. There's sometimes just a short delay before she can speak because it just needs to recalibrate the system.
Okay.
Jy Beckner, jy is jou wireless of kan jy nie inplug via die metabolome.
You're not on the TV? I see.
Sam is still lit up.
Okay.
We want it to at least function and cool down a bit, but yeah. There is no. What? That people will not complain. Is that woman's brave enough to take on the heat in this room? Okay, Megan, the jokes here is definitely on the heat that we're experiencing. They're not laughing because of the presentation still to come. So I think, are you ready?
I am.
Okay. So good morning and welcome to your big day where you will be defending your PhD. Megan, as you see, has chosen to rather do her defense via Google Hangout, which she challenged us a bit with in the week, and rather fly out to come and get her degree hopefully in March after the she decided that for her all over the world, as far as I understand, somebody in China and the parents in Italy.
Yes.
Okay, so, and then obviously most of Megan's PhD was through the summer school, which Justin was also in San Michele with Fondazione Edmund Mach. And to the people from FEM who's also listening, Ushka as her co-supervisor, where she did most of her work, welcome to you. And I assume there's quite a few other people in another room listening and tuning in to YouTube.
Yes, there are people, some people also from San Michele who are listening in defense now, yeah?
So yeah, so Megan did a master's at in the States where she actually worked on something quite very related to her PhD. She started off with looking at volatile compounds from Saccharomyces and Lactobacillus plantarum, which I actually only realized when I read your biographical sketch that the overlap in what we do here and what you had as previous experience was actually very relevant. So, as I say, she was part of the PhD program in San Michele, and as part of that, because it's an international program, I remember that Fulvio called me, I think on the 30th of December, to write up a proposal to submit this project to their committee, and I presume that nobody was at that time really interested in spending a lot of time, so we got the program approved. And Megan then applied and was the chosen candidate to do this. So Megan then worked on metabolic profiling on non-Saccharomyces yeast in wine. And obviously, between the different environments where she spent time, she got a lot of information and knowledge. And all this knowledge and huge data set, she will now present to you in defending the work that she's done in the last 3 years. So Megan, good luck. We look forward to your presentation, and obviously this for us is the first experience doing it online, so I hope the internet doesn't disappoint us in this case. So Then afterwards, just logistically, if you have any questions, you will have to come to the computer in front or shout from wherever you are. So, otherwise, she can't hear you. Can you hear us?
Yes, I can hear you very well, Margaret. The room as a whole does echo quite a bit, so anyone further back, it is going to get a little tricky. You'll just have to speak very clearly, or as she said, I think come up a little closer to the microphone.
Okay, so good luck. We look forward to your presentation.
Okay, well, thank you, Margaret. I'll very quickly be switching over to the presentation, and you will just see that and hear me. I can see all of you during this. I would ask if the quality gets very, very poor for someone to either raise their hand or jump in and say so that I can pause so that you don't miss anything. So with that being said, thank you again, welcome, and here we go. Okay, so as Margaret said in the introduction, I will be presenting my work on metabolomic profiling of non-Saccharomyces yeast in wine. I am Margaret Elizabeth Beckner Whitener. Today's February 1st, and we are presenting over the internet at the Institute for Wine Biotechnology in Stellenbosch, as well as my advisor at Fondazione Edmund Mach in Italy, while I'm in the US currently. So I'd like to begin by going back to October of 2012. As Margaret said, I graduated from North Carolina State University with a master's in food science, where I had been working on Saccharomyces yeast and interactions with lactic acid bacteria. I'd done a little bit of work chemically speaking. The majority of that work, however, was microbiology and genetics related. So I was very excited when there was a project posted at Fondazione Mach entitled Mass Spectrometry-Based Metabolomics of Non-Conventional Yeast and its Application in Enology. So I applied and was accepted and started in October of 2012. That title very quickly got changed to something that's a little shorter, a little more concise, and the project as a whole took on the focus of non-Saccharomyces yeast in wine and metabolomic profiling therein. So to really begin, I'd like to give an introduction and go through some of the project goals. The project had been written, basically a brief outline had been written by Margaret and by Fulvio, by Erska as well, and one other collaborator at the University of Lund. And my first job was to sort of distill that down into something we knew we could tackle in 3 years. And to do that, I needed to understand what's called the current state of the art. What do I mean by that? Specifically, I needed to understand what we currently know, or knew at the time, about the role of non-Saccharomyces yeasts in wine. And I'll pause here to give a brief background. Saccharomyces cerevisiae is the yeast primarily responsible and used in alcoholic beverage fermentation, whether it's wine or beer or alcohol production in general, because it is such a good yeast at producing alcohol. That being said, there are quite a few yeasts specifically related to wine that are known to be present on the grape berry in the winery environment. And these belong to, as it turns out, tens of different genera, which encompass possibly hundreds of different species. So that was goal one, understanding what we know, how many yeast there are, and what do we currently know about how they impact wine flavor and aroma. The next step was to define for myself and to understand what metabolomics meant in this context. It's a very broad field of study. The technical definition, I suppose, is the scientific study of a set of metabolites present within an organism, a cell, a set of tissues, or just a system in general. Specifically, I sought to understand what kinds of technologies are currently being used to profile the metabolite production of a given organism. And a lot of different technologies are currently being used. Chemical, genetic, microbiology, it's an all-encompassing field. So with all of that understood, goal 2 was then to develop analytical methods to consistently and accurately characterize the volatile profile of wine. And this was one of the first points that I really had to narrow the field on. Again, metabolomics being such a huge field, we chose to focus just on the volatile metabolites that were being produced by the yeast in the wine, since those metabolites ideally are the ones that are contributing most to flavor and aroma. If they're volatile, we're going to be able to smell them, we're going to be able to taste them in the wine. So to do that, we had quite a few platforms at Fondazione Mach in the metabolomics platform, and we were able to come up with a targeted and both an untargeted analytical method, which allowed us to have a certain set of metabolites with regards to the targeted analysis that we could consistently identify and also quantify. So we picked out some relevant that are some of the most known to be relevant in wine. And then with the untargeted method, we wanted to be able to take the biggest and the broadest look that we possibly could to see what's going on in the wine. The next goal, once those methods had been developed, were to then characterize the behavior of select non-Saccharomyces yeast in both a red and a white grape must. It was important to us to understand if there was in fact a difference in how these yeasts respond to a red and white grape environment. This was done on a very small scale, and the idea being that if we got relevant and good data, we could then scale up. So the next goal, the next phase of the project would be to produce full wine from start to finish. Once the wine had been produced, that's where a lot of those chemical methods and analysis techniques come in. And we were also lucky to be able to collaborate at Stellenbosch with the sensory department and do sensory analysis on our finished product as well. This, of course, created a lot of data, and so the next goal was to figure out the best ways to analyze and make use of that data, to correlate the chemistry and the sensory data, and get the most out of what we had produced. And then the last goal of the project as a whole was to publish the work, obviously. We were actually very lucky. We've had 2 papers accepted, and we should be submitting one more very shortly. So now that I've given you an overview, an idea of sort of where we're going, I'd like to dig into the details and show you kind of how we got there. First, the analytical method development. If you'll recall, the goal here was to develop both a targeted and an untargeted method to characterize the volatile profile of wine. So to begin with the targeted method. In a lot of ways, this one was actually much easier to develop. We had several methods in-house that had been used on fermentations, and essentially we just needed to add to that to be able to characterize wine to its fullest extent. And the way that we did that was with a GC triple quad mass spec. GC stands for gas chromatography. It's a system that utilizes one separation column, and then in this case, if that column, once the analytes have been separated, will then pass samples to a triple quadruple mass spec, which is unique and very useful for targeted analysis because, as you can see here, a sample will come in to the analyzer, it will be ionized, and then it goes through several stages of filtering. The first quad is responsible for filtering just by the mass, and this is completely chosen by the user. So if you know what you're looking for, you can set your parameters to get very distinct profiles, which is great. So you select your mass. It then goes to the second quad, which is another collision cell, which essentially further breaks your ion down into more fragments. And then those are further separated before they finally hit your detector. And for anyone not familiar with what this data looks like, you can see here an example. We've got typical gas chromatography spectra on the right, and or I believe it'll be left for you all. But you'll see that highlighted here. Essentially, each of these peaks, ideally, that are numbered, would represent an individual compound. So this is the separation over time on that GC column. And then after it's been broken up into its constituent pieces, what you end up with is this mass spectral graph. And we are very fortunate to have a very robust set of libraries that have cataloged the spectra of huge numbers of chemicals. And this one here, for example, is ethyl acetate. So we know if we see this pattern that, in fact, that peak is ethyl acetate. And we can do that for each one of these. So moving on to the untargeted method. The idea here being to see as much as we possibly can. We were fortunate at FEM to have a new GC-GC time of flight mass spec, which utilizes, obviously, GC-GC. It's got two chromatographic columns in it. And you see an example here in the image where a sample will come in to your GC and pass through the first column, and analytes get separated, just as they would with the targeted method. And then the analytes are passed into a second column, which is a different phase. And you see that here, this smaller column. The purpose here is if, and this is very common in GC, you can have a single peak within a system which actually, because of a lack of separation, encompasses two or more analytes. So with GC-GC, you can get two dimensions of separation from two different types of columns, and then samples get passed off to your detector. In this case, a time of flight mass spec, which instead of all of those different collision stages, separates out your ions based on the amount of time they spend in your flight tube before they hit the analyzer. And you still get those, the mass spectra, the nice clean mass spectra, which you can then compare to the databases and see what's going on. This type of data does look a little different, again, because you have two columns that are doing your separation, but the end result is that you get dramatically increased peak capacity. You get, obviously, improved peak resolution, assuming you've selected the right types of columns to do your separation. And you can get up to an order of magnitude increase in total compound detectability, which makes this platform ideal for an untargeted type of analysis. So we're going to put all that on hold, keep that in the back of your mind, and I want to move forward and talk a little bit about yeast strain selection and the microfermentations that I did. So the goal with the strain selection was to characterize the behavior of select non-Saccharomyces yeast in red and white grape must as a sort of a preliminary approach to get an idea before we went into full-scale winemaking of what was going on. A brief introduction to go back a bit. Saccharomyces cerevisiae, as I said, is the primary yeast responsible for fermentation because it produces alcohol so well. What's interesting, and we know from the research in the literature, is that it typically only accounts for 1% or less of the total yeast population found on healthy, undamaged grape berries. And if you pause and you think about that for a second, it sort of might seem like a miracle over the thousands-year history of winemaking that we ever even found Saccharomyces cerevisiae in the first place, right? That this yeast, which is hardly even there at all, would become the dominant yeast for fermentation. And that has to do with the fact that, again, Saccharomyces is just so good at making alcohol. Even if it's present in very, very low quantities on the grapes, what happens is once it gets into its niche environment where it's got enough sugar, it starts to grow and produce alcohol. And alcohol, as it turns out, is rather toxic to most living things. And it just very quickly is able to outcompete the rest of the yeast that are present. But what about those yeast? If grape berries, and again we do know this, are dominated by these low alcohol tolerant yeast species, even if Saccharomyces takes off and runs the show, they're still there. They're still metabolically active. And again, we know from literature and from the previous research that there are, as I said, tens of genera encompassing hundreds of different species of yeast that have been found to be present on grapes. And a typical grape fermentation, if it's not aided, so if yeast are not added to the system, you'll see sort of this progression where species of, for example, Cloagra and Hemphanaspora, which are very, very low ethanol tolerant, will start everything off. And then you move into some of these others, the Metschnicovia, the Pichia, the Trilospora, the Shantia. Those tend to take over. They're slightly more ethanol tolerant. But eventually Saccharomyces wins out, and these just are no longer really present. And so that's where we chose, and again, this is mostly due to literature research, to focus our efforts. Not only because these yeast are slightly more tolerant, meaning they're probably going to stick around a little longer in the fermentation and have the most effect overall, but because, as it turns out, the Metschnicovia, Pichia, Trilospora, and Lachantia genera have a couple of species which are actually being produced already and made available to winemakers, because there is some preliminary data to suggest they can enhance the overall flavor, aroma, or quality of a finished wine product. If you'll remember back to the introduction, I said that we had a collaboration with the University of Lund up in Sweden. They were a yeast lab that were associated with the Cornucopia project and had done some research which indicated that species of Zygosaccharomyces and also species of Kazakhstania, while not typically present in a winery environment or in a winemaking environment, could in fact have some interesting volatile characteristics. And so we went ahead and chose to throw those into the mix as well for our initial testing. So the setup, how did I actually produce the data to get a preliminary look at all of this? Well, again, chose these species that you'll see over here on the left. Saccharomyces cerevisiae was used, obviously, as the control. And then we had Zygosaccharomyces kombuchensis, Kazakhstania gammaspora, Trilospora delbrueckii, Lachantia thermotolerans, and Metschnicovia pulcherrima. These three at the bottom actually were the commercially available strains, because we wanted to get an idea more specifically about what these strains were capable of, because there really just, as it turns out, isn't a lot of data, despite the fact that they are being commercially made available. So harvest in Italy of 2013, we had Syrah and Sauvignon Blanc. I was given these musts to work with and came up with a system in which we put about 55 milliliters of must into a vial, very similar to the design that you see here, capped with this little a filter at the top, which was sterile, which would allow for gas exchange. And the yeast were inoculated into those vials and 3 biological replicates. And those yeast were allowed to ferment until they reached a 2% ethanol concentration. Why 2%? Well, as I said previously, a lot of these yeast are very low ethanol tolerant species. Each of them does have their own threshold, but 2% is concentration in which we knew they could all reach, because they all are capable of producing at least a small amount of alcohol by themselves, and it's the point at which, for some of these, that environment may start to become toxic. So once the yeast had reached, or once the fermentations had reached that 2% mark, the samples were spun down, the supernatant was taken off, and that's what we analyzed. And we used SPME, solid phase microextraction, to do the extraction of the volatiles. And essentially what you do there is put your media into a vial. A needle is inserted, which has a coated fiber on the tip. And that fiber is coated with, or can be coated with, various things that will absorb the volatiles present in the headspace. That fiber is left in contact with the headspace for a given amount of time before it is passed to the GC inlet and desorbed. It's superheated under a gas stream so that it is pushed onto the GC column so that analytes can be separated. We used, in this case, the targeted method that we had developed, which allowed us to target 90 different compounds total. Not only target, but semi-quantify in this case. The sample was spiked with several internal standards, and one was chosen to compare its relative peak area width to everything else. So we could get an idea, relative to the internal standard, approximately how much of each of these compounds was present in a given sample. And I'd like to highlight and draw your attention at this point to the alcohols, the esters, and the terpenes that we analyzed in this system, because arguably these 3 classes of compounds are responsible for the majority of flavor and aroma in a wine. You can see here that alcohols in general have green, sweet, fruity, apple aromas. In too high a concentration, they can also contribute to fusel, which can be an off flavor. Esters, on the other hand, and these are arguably the most important, give you all of those really great fruity characteristics. And you can see a few of them listed here. Terpenes are interesting because they're naturally present in the grape, and rather than be produced directly by yeast during fermentation, like alcohols and esters typically are, the yeast will produce enzymes that are able to liberate the otherwise bound terpenes and make them sensorially available to us. And we'll get the citrus, the rose, some of these deeper, woody, even licorice aromas and flavors out of the wine, thanks to the presence of the yeast. So with all of that in mind, let's go ahead and take a look at the data. What you see here is the principal components analysis for the Sauvignon Blanc and the Syrah. And we were very excited to see that in total, about 60% of the variability that we saw between all of the species was represented here, and that each of the biological replicates grouped very well with themselves, but were obviously very, very different from each other. And I'll go ahead and highlight some of the main story here. If you'll notice that the first factor represented over 40% in the Sauvignon Blanc and 35% in the Syrah of the total variability. And we see that in this case, the Saccharomyces cerevisiae control and the Zygosaccharomyces kombuchensis fermentations differed the most significantly along that in both lists. And then we see that the second factor while only about 20% of the variance is explained by that, we see that the Kazakhstania is up there as well, and that is these differences and separations are further confirmed by the hierarchical clustering analysis that we did. So we see here that the yeast themselves grouped more significantly together, regardless of which must they were present in, which in and of itself is very interesting. And then we can further see, so just to drive that home, we see here that the Saccharomyces in the Sauvignon Blanc and the Saccharomyces in the Syrah, you see them highlighted in the tan and the red, were the most similar to each other compared to everything else. And that trend continued for all of the fermentations. We see the first major branch in the hierarchical clustering is the Kazakhstania Gammospora. And then we see the two most extreme ends represented by, again, the Saccharomyces on one end and the Zygosaccharomyces on the other. And for the sake of time, I'm really just going to talk about these three yeasts because they were the drivers of the main story, I would say, from this data. When we further break everything down into its constituent pieces, so to speak, and again, I'm just focusing on the alcohols, the esters, the terpenes, those things that are going to be driving the majority of the flavor profile of a wine for the sake of time. What we can see in these box plots, again, we were able to quantitate the individual compounds, and what we see here represented is the relative total of the compound classes. So the relative total of the alcohols and the relative total of the esters represented here. And I want to go ahead and just walk you through this data briefly. The Sauvignon Blanc data is represented on the left-hand side of the graph, while the Syrah data is represented on the right. We can see, and these are always going to be in the same order. It goes Saccharomyces, Kazakhstania, Leschantia, Metastrachovia, Triospora, and Zygosaccharomyces down the line for both the red and the white. So in the case of the alcohols, what's the story here? Well, we see that overall, the overall concentration of alcohols was highest for the Saccharomyces fermentations. We see that, relatively speaking, the lowest was actually the Kazakhstania. And again, just because these were the main story, the Zygosaccharomyces was sort of somewhere in the middle. And the same relative trend, although total concentrations were lower, was seen in the red. Jumping over to the esters. Again, we can see if we just draw this line here, that the overall profile in both musts was approximately the same. Further giving this idea that regardless of the musts, the behavior of these yeasts is approximately the same. This one's interesting because the Kazakhstania was the one that in both musts produced the most overall total esters. Close behind it was the Saccharomyces, and then the Zygo did basically nothing. It's right there near the 0 mark. We can say it basically produced almost no esters to speak of. Moving on to the terpenes. This is where things get interesting, and we can see that it's always relevant to dig deeper into your data, because the overall trends are telling us that these yeasts are behaving the same regardless of the grape type that they start with. And yet, taking a look at the individual compound classes, we can see that that's not always the case. If you'll remember, terpenes, as I said, are not produced by the yeast themselves, but rather liberated from the must. And so if we were going to see a difference between certain profiles, we would actually expect this to be it, because Syrah has more terpenes total to work with than Sauvignon Blanc. And that is the majority of the reason why we see such a difference here. In general, we can say the Saccharomyces produce the most, well, very closely followed by the Lachantia in this case, but the most terpenes in the white. Slightly different story, obviously, over in the reds. And I'd just like to point out that regardless of the must, the zygo, again, similar to the esters, did a whole lot of not much. I do want to point your attention to, briefly, the phenols. And I threw this in here to be able to illustrate a point of, because, you know, with these fermentations having been so small, we weren't able to do a sensory analysis. All we can go on is the chemistry data. And you would think that, oh, well, the more a yeast can produce a given class of compounds or a given compound, the better. You know, the more fruitiness we can get out of our wine, the better. The more, you know, grassy greenness or the more tropical notes out of a Sauvignon Blanc we can get, the better. That is unfortunately not always the case. There are classes of compounds which, when present in low amounts, can be very beneficial, can contribute to the overall complexity of the wine. However, once they reach a certain threshold, they become off-putting. And phenols is a great example of that. In low concentrations, they can give these rose and clove and spicy, smoky notes, which can be great. Too high a concentration, you start to get things like sweat or medicinal or barnyard, things that nobody really wants in their wine. And so that's something to be careful of and something worth noting. We can see here that in Sauvignon Blanc, the highest producers were, again, the Saccharomyces and the Lachantia, with zygo producing little to none. That trend was slightly different in the Syrah, with the Sac producing relatively, you know, something in the middle, and actually the Kazakhstania being, even though that variability was pretty high, being somewhat higher overall. So to summarize this data, and again, there was much more that I could have gone over, but for the sake of time, I gave the overview. We can say from this set of experiments that non-Saccharomyces yeast have a potential to play a positive role in winemaking. We were able to further conclude that the majority of flavor compounds, the alcohols, esters, and those terpenes, were produced in lower concentrations by the non-Saccharomyces yeast than the Saccharomyces. The exception being the Kazakhstania gammaspora, which actually produced higher overall ester content than the Saccharomyces control. We also further concluded that going forward, the Zygosaccharomyces was likely ill-suited for increasing wine aroma complexity, if for no other reason than it really just didn't produce much of anything compared to some of the other species. So with all that data in mind, we felt confident going into the full scale up, which took place in Stellenbosch in harvest of 2014. The fermentation method for this was, again, Sauvignon Blanc and Syrah. In this case, we had, again, Saccharomyces as our control. And again, we had the commercially available strains, which you see blocked off there. And then the zygo, as I said, was not, not something we felt we needed to further investigate. And unfortunately, we couldn't get the Kazakhstania gammaspora down to South Africa in time. They happened to have on hand some Kazakhstania arabica, which had not been tested in wine to this point. And from some preliminary data as well, and from some literature research, we went ahead and threw in a Candida zemplenina as well. So these were the chosen yeast, and rather than in the very, very small scale, the 55 milliliters, I actually did 10-liter fermentations. Again, 3 biological replicants were used, and again, that 2% ethanol cutoff was used. But because we needed to make, wine fermented to completion. That 2% ethanol mark served to be the point at which we added Saccharomyces cerevisiae to the non-sac fermentations so that the rest of the sugar could be fermented and we would have a finished wine product. In this case, the analysis that we used was, again, a speedy extraction, but we wanted to get that big picture. We wanted to get the everything. So we used the GC/GC untargeted method to do the analysis. And you'll notice here at the bottom, I have 3 different times listed: 10 seconds, 5 minutes, and 30 minutes. And those times represent the total extraction time. So the total time that the fiber was left in contact with the headspace of a given sample. And the reason for that is, as I went through the method development, which was very, very interesting, but very complex and very difficult with the GC/GC, we noticed very quickly that you would very quickly, because this method is so sensitive, get to a point of saturation in which your peaks could no longer be reliably integrated. And what I mean by that, if you'll see here, we'll follow compound 1 here. We can see that at a 10-second extraction time, it is this gorgeous peak shape. This almost perfectly round little dot, which the computer then has absolutely no trouble integrating the same over and over and over again. So you could run, 20 samples that all have this compound in it at this concentration, and it's going to tell you that the area under that peak is basically the same regardless. 5-minute extraction, if we keep following compound 1 here, you can see we're already getting some tailing in the second dimension. And the more tailing you get in GC in general, the more difficult it's going to be to calculate that area under the curve. But this is especially difficult in 2D GC because, and you can actually see this in the software, if you were to turn all this on its edge, these aren't just peaks like we're used to, but they're actually cones. They're represented in 3 dimensions. So you're actually having to calculate the area under a cone, not an area under a curve. And that gets tricky for anybody, even the computer. It's especially tricky when it just becomes that much more blown out. And what you end up with is inconsistency in the total calculation. So if you want to get the most out of your data and you want to get everything that you possibly can, I made the decision to go ahead and do these extraction times sort of staged, because as you can see, the example of compound number 2, there are some compounds which you're only going to see at those longer extraction times. That needle just has to be in contact with the sample for longer to pick up that compound, either because it's just not present in a very high concentration, or it doesn't bind as well to the fiber matrix as some of the others. So with that in mind, I'd like to go ahead and share some of the Sauvignon Blanc data. And I'll start with the fermentation progress, which is interesting for a couple of reasons. You'll see here that ethanol production is represented in red. Glucose consumption is represented in green. Fructose consumption is represented in blue. And you'll see the vertical lines on the graph. In the case of the non-Saccharomyces fermentations, which start here with the candida and move all the way down, this solid line is the point at which 2% ethanol concentration was reached. So that would be the point at which that the Saccharomyces was added to ensure the completion of the fermentation. And then, because these were not only tracked chemically, I also tracked everything by plate count. The dotted line represents where those non-Saccharomyces yeast species were no longer detectable via plate count. They had been overrun by the Saccharomyces. So we can already see that some of these fermentations did behave differently from each other. One difference I want to draw your attention to is in the candida. where we can see that unlike the rest of the fermentations, fructose, again, which represented in blue, was actually preferentially consumed throughout the fermentation. Its line is going down faster than the glucose, which you can notice is not the case in the rest of the other fermentations. Now, we do know that Candida xanthoina is a fructophilic yeast, so we would have expected, at least at the beginning, when that was the dominant yeast in the fermentation, for fructose to be consumed over the glucose. What's interesting is that trend continued throughout the fermentation. Even after the Saccharomyces was added, even after I couldn't detect the Candida via plate count, we can see that fructose was still being consumed more rapidly than the glucose. So that was one of the interesting things before we even dive into the chemistry data about the differences of these fermentations. So getting on to the chemistry data, the GCGC method is robust. And not only did I have 3 different extraction times to work with, but each one of those presented over right around 1,000 on average individual peaks. So you see that represented here in the hierarchy of clustering. The 10-second extraction put us at 988 ANIs. The 5-minute, we actually had 1,500. And the 30-minute extraction were right around 1,000. And so step 1, when you've literally generated a mountain of data, is to get the big picture view of it, right? To know whether or not it looks good. And one of the reasons that we did the hierarchical clustering you see here is to check and make sure that our yeast treatments are grouping together nicely, and we did see that. We're also looking for, in this case, where the story is with our QCs, which you see in pink. QCs in this case were pooled samples, which will become very important later. Common standard practice in chemical analysis, especially of this type, in that the sample tray is set up with your samples randomized. In our case, we did every 4th injection was a QC sample that ideally had equal parts of all of the other samples mixed into it. And so you do want to see right off the bat that, and you can see this very easily in the graphs, that the QCs, the overall intensity of all of those peaks is much lighter because it's a dilute sample, right? If you've got a mix of everything, no one thing should be dominant. So that was very good to see. But these graphs don't tell us much more than that. To get at the heart of the data, we had to come up with a way to filter it. We had to drill down into what was most relevant. What's driving the separation of the hierarchical clustering? And we came up with, I worked very closely with the chemometrician at FEM. We came up with a method to try to filter out some of this, and it was a 3-fold filtering process. We compared all of the yeast to each other and said, okay, we only want to look at, for any one comparison, any compound which had a 2-fold change or higher, just based on the peak intensity. The second, so that represents that fold change filtering here that you see in the purple. The second criteria was based on the QCs. Now, if you remember, I said we were seeing that if a peak wasn't shaped very well, the computer might have trouble integrating it consistently. What we did here is we used the QCs to kind of help mitigate, even with the 3 extraction times, you're still not going to get perfect peaks all the time. So we said, okay, well, we only want to look at peaks that we know were integrated well. How can we do that? I can't sit there and check every single peak integration. That's unrealistic. So we said, okay, well, let's look at the coefficient of variance within the QC samples and choose compounds, because ideally in a pooled QC, all of your compounds are there. Choose compounds which had a variance of 50% or less. And so that was the second filter criteria. And the third was by Q value, which is calculated through a false discovery rate algorithm to further mitigate any possible false discovery, any false positives, essentially. So we selected what could be called a very narrow set of criteria. We took the data from thousands of possible analytes in the 10-second extraction, 78, in the 5-minute, 196, and again, this is for the Sauvignon Blanc data. And in the 30-minute, we had about 239, what we were then referring to as compounds of interest. The things that were ideally driving the majority of the differences between our fermentation treatments. Taking those and plotting them with PCA did in fact reveal that there was some pretty distinct separation between our species. And we were fortunate, like I said previously, to work with the sensory department at Stellenbosch, and they evaluated the Sauvignon Blanc. And we were really, really excited to see that our chemistry data, the differences, the major story that we're seeing, also showed up in our sensory analysis. And of course, I'm referring here to the difference between the Saccharomyces controlled fermentation and the Candida fermentation, or mixed fermentation. For the sensory data, I want to briefly go over that. We can see that, according to the plot here, the major drivers for the control, the Saccharomyces, were guava, passion fruit, grapefruit, pineapple, banana, all these great, everything you expect in a Sauvignon Blanc. Those great tropical flavors were the dominant flavors for the control. On the opposite end, we see that while guava, being at the top, still pulled the Candida mixed fermentation up, it was much more dominated by fermented apple aroma and some of the more dried or cooked fruit flavors, which is very interesting. And if we then go over to the chemistry data, if we then go over to the chemistry data and we look at the chemicals that we had found, our compounds of interest, we saw that the Candida fermentations were producing significantly more terpenes in general, and then there were certain specific esters that it produced a lot more of than others. But in general, the control still produced the most total esters, I would say, and alcohols as well. So again, we're sort of back to that story of it produced the things that are the most things that do contribute to wine flavor and aroma. So to summarize briefly the Sauvignon Blanc, we concluded that the major story was the Saccharomyces versus the Candida zephyrina, which had the most distinct and remarkable fermentation profiles, both sensorially and chemically, and that surprisingly our sensory and our chemistry data sets complemented each other really, really well. Overall, from this, we can say that the non-Saccharomyces yeast, that while they produce wines that were unique, still the Saccharomyces in single culture produce the strongest positive sensory components, mostly due to its high ester production. So to move forward, if you remember, I did not only the Sauvignon Blanc, but Syrah as well. It followed the exact same protocols from fermentation all the way to data analysis and beyond. With the exception of the fact, you'll see here, I've only got the 5-minute and the 30-minute extraction time data are represented in the PCA. The compounds of interest are represented here. And that's because overall, Syrah is a much less volatile grape and much less volatile wine. And it turns out that 10-second extraction didn't actually give us any useful data. There just wasn't enough there to be worth anything. But again, the 5-minute and the 30-minute still gave us over 1,000 analytes each to deal with, and had to be further filtered to get at the differences. I was kind of shocked initially. If you'll remember back to the preliminary fermentations, we said, well, in general, the profiles are the same, regardless of the yeast, or regardless of the must that you start with. That very obviously is not the case if you have a finished wine product. Here, rather than Saccharomyces and Candida, the major story is Saccharomyces versus the Lichtenstein thermotolerance. We were unfortunately not able to do sensory analysis on these wines. The timing just didn't fit. But the chemistry data shows us that the Lichtenstein was unique for specific production of certain esters, acids, surprisingly, and terpenes, which actually correlates well with our preliminary study, where we did see the Lichtenstein fermentations produce more terpenes, especially in the Syrah, than even the control. And we also saw, and this was unique to just the Lichtenstein fermentations, a single compound listed here. It's a pyrrole, which further research, literature research, revealed that this particular compound has a sort of spicy or smoky flavor or aroma, which is something that's very desirable, actually, in Syrah. So it was really interesting to see that only one yeast fermentation produced this compound, which is potentially very beneficial to Syrah, sensorially speaking. So to give some comparisons and further conclusions, in the Sauvignon Blanc, the major story was between the Saccharomyces cerevisiae and the Candida zemplenina fermentations. In Syrah, the major story was between Saccharomyces and Lichtenstein. And we can very clearly say that the differences between the species, or the species did behave differently depending on their starting matrix and their fermentation conditions. But clearly, more research needs to be conducted because we really only scratched the surface. And that brings me to possible future work. The techniques that we used were state-of-the-art. And yet, we couldn't actually really concretely answer the question of which compounds, or indeed class of compounds, were responsible for the fermented apple flavor, for example, in the Candida Saccharomyces co-fermentations of the Sauvignon Blanc. We gave a comprehensive chemical analysis, but if that were to be combined with, for example, genetic expression data, we could really start to get at what I think is the core of metabolomics research, which is really understanding not just, metabolic output, but the processes that provide them. The data, as I mentioned, was actually filtered very, very conservatively. And so it's possible that we missed some really interesting or relevant points. To that end, we have made the data publicly available on the metabolomics repository. I'd actually love to eventually go back or work with somebody to maybe tackle it from a different perspective and see what else this data may have in store for us. We obviously weren't able to track any of the non-volatile constituents, either their loss or their production. And this, of course, would help us to further form a more full metabolic profile for each of these species, especially now that we have an idea of which of these species are potentially going to have the most effect on wine. We could narrow our study and do a much more in-depth study of a specific species rather than take this broad. Finally, obviously, all of this and more can and I think should be done to study the role that these species have to play in the future of the wine industry. So to bring it all back around, I started in October of 2012. It's now February 2016. And in the course of that time, oops, I hit that slide, sorry. In the course of that time, I was able to show that non-Saccharomyces yeast can consistently and dramatically impact wine chemistry, flavor, and aroma. I was able to create a comprehensive profile of the volatile chemical impact of 7 different yeast species in 2 different lines. I was able to show that the yeast behaved differently depending on which grape environment they were grown in. And from this data, I published 2 papers, and I expect to be submitting another one in the next month. I'd like to take this opportunity to acknowledge my funding bodies. Fondazione Edmund Mach, the GNPF program, was the main support, and then our collaboration with our other universities was funded in part by the grants listed here. I'd like to give a special thank you to the Metabolome Lab at FEM, of course the Institute for Wine Biotechnology there at Stellenbosch, the Piscor Lab up in Lund, and I really couldn't have done this without my colleagues, my friends, and of course my family. They were an amazing support structure, and overall I can say I actually had a wonderful time doing this project and learning everything that I did. And so with that, I will take any questions.
Thank you, Megan, for a very interesting presentation. And I know the data has been complex, trying to simplify it for people to understand it. So yeah, as Megan said, the floor is open for questions. I mean, obviously, you are welcome to also ask questions from your side. So whoever is first, come to the computer, please. Florian?
Megan, can you hear me, or do you have to? Can you hear me? Oh.
Not quite. I'm sorry. It's very echoey. So the further back you are, it just gets garbled.
Hi, Megan. Can you hear me now?
Yes, I can. Thank you.
Megan, in your conclusion and during your data, you speak about species. Now, you use the single strain for each species.
True.
And you know that there's quite a range normally of all of those species.
There is.
Would you think that these profiles that found are outside of the species space of Saccharomyces alone? Can you discuss this a little bit? Saccharomyces alone has a huge space.
It does.
Do you think that what you see is outside its own would be able to? Can you discuss this a little bit, this issue?
I can, and I can actually tell you that from the literature research, there have been several studies that have looked at just a single species of a given yeast, either it's Saccharomyces or a non-Sac, and looked at strain variation. And you're right, we do see a pretty significant difference in some cases between even individual strains of a given species. And that would be one thing I probably should have added to future work or to follow up. It would be great to better understand, even within Saccharomyces, how those different strains are really producing different flavor profiles, and again, why. I really wish we could have done a genetic component in this project, because there is, the possibility that even one slight change, not enough to be considered obviously a different, even strain, in some cases, could really significantly affect a volatile flavor profile. So I hope that sort of got at it. If you'd like me to explain further, I could.
Just another small question. How did you handle oxygen during your analysis? I refer in particular to the Semplinina data and the glucose-fructose concentration. Shown that this is happening in particular when you actually make oxygen available throughout fermentation, because then Candida is actually able to consume fructose a lot more efficiently.
Right.
How was your oxygen management?
So if I understand the question correctly, because it did drop out, you're wondering how oxygen was handled in the large-scale fermentations, correct?
Yes.
Okay. So in the Sauvignon Blanc fermentations, they were actually handled slightly differently. Obviously, in a red wine, you do punch downs, which is going to introduce oxygen into the system, more so than you do for a white wine fermentation, which is left completely static. There was no oxygen added So the only oxygen present for the white wine fermentations would have been that was available in the very small headspace, because we did have the 10, I suppose there were overall 10.5 liter canisters, but it was 10 liters of must. So you did have a little bit of an oxygen headspace, but that was it. There was no mixing, there was no addition of oxygen to the system for the white wine. Again, with the red, which was done in the same canisters, because you're doing those punch downs, you do have more oxygen available.
Okay, thanks.
Quibbers? Those who have questions, maybe you should line up in a row so that you don't waste time. So. Well, Megan, I must say you've achieved something that's never happened in the meetings before. You have the people who ask the question on their knees. Normally it's the other way around.
Oh, the magic of technology.
Hey, Megan. We discussed some of your results last year when you presented here. The first question I have is your data on the terpenes. You did mention the concentration of the terpenes you had. Was any of the levels of terpenes that you analyzed there close or above the odor threshold that you can expect in a wine environment? You can ask that first, and then I'll ask the second question.
Sure. So the only data that we quantified for terpenes was the micro-fermentation data that we used the targeted method. And in that, we had, I believe it was 17 different terpenes that we were able to semi-quantitate. And yeah, 17. So whether or not they were above the sensory threshold, if I recall, and I will have to double check, the answer is no. But that's actually not surprising given that the micro-fermentations didn't go to completion. So again, remember, we cut everything off at that 2% ethanol mark, which meant, the fermentation wasn't even half done, essentially. And I wish we had been able to go back and do a quantitative analysis on the finished wine. There really just wasn't time. And so, fortunately, we did have, of course, the sensory analysis. But I can't speak specifically about whether or not the terpene content in the finished wine products was at or above the threshold beyond what the sensory data was able to tell us. So.
Okay. And then the second question is maybe a little bit more philosophical, and you mentioned it at the end with when you said your future work and what was maybe a shortcoming of the study. And the question is, it's production versus liberation. You said that Shiraz was a lower volatile cultivar. And my question is it possible that the difference that you see is mainly due to having a more conjugated content of, say, terpenoids in this case, in Shiraz versus Sauvignon Blanc? And then to what extent do you think the different yeast strains are able to liberate these compounds from the must environment?
Right.
You also said that they produce these terpenoids. I'd like you to elaborate on that, whether this is from a grape-derived precursor or whether they do this de novo. And then if there's any information on the type of enzymes that these potential yeast strains produce that could liberate terpenoids.
Right. Really good question. And I delved into the subject a lot in the research chapters in the thesis. To briefly summarize, to my knowledge, and of course I could be wrong, but to my knowledge, de novo synthesis of terpenes hasn't been found in the strains that we studied, but enzyme production actually has to some extent been studied in some of these yeast, and we do know from previous data that certain yeast are, and especially non-Saccharomyces yeast, this is, I would say, one of the reasons why these yeast have really become so interesting in recent years, is because they are capable of producing a pretty wide variety of enzymes that Saccharomyces in and of itself just is not. The glucosidase enzymes, so the most common being beta-glucosidase, for example, has been shown, and I do believe it is in Lachantia, as well as some Candida strains. They've shown to produce quite a bit more than a lot of other yeast. But it is sort of getting back to one of the other comments of it's very strain dependent. So, again, further studies should be done that would more specifically characterize, and possibly not only genetically, but expression of some of these to really try to understand what's going on there.
Cool. Thanks, Megan.
Okay. Evodia's next.
Hi, Megan.
Hi.
I have a quick question for you. If you look at your chemical profile of the non-Saccharomyces yeast, you have quite a few of them that are not really distinct from each other. So what I would like to know is how different were they in terms of the dominant non-Saccharomyces yeast at the point of inoculation? So at the 2% ethanol point, did you analyze the community? Do you know how distinct the community was from each other?
I assume you're referring to the full-scale wine production, or just everything in general?
No, the full scale.
The full scale. So the point at which, because I did sample pretty much daily throughout these fermentations for the chemical, the base chemical, so glucose, fructose, ethanol, all of that with the FOS, and then the samples were taken to run on plates. Prior to Saccharomyces addition, the fermentations were more or less evenly dominated by the inoculated non-Sac strain. There were a few, I do recall a few colonies of other yeast present, but the inoculation levels were even for all of the yeast, and they grew at approximately all the same rate, actually, which is kind of surprising. From my preliminary study, I kind of expected a few to lag behind a little more or less, but I was actually very lucky that they all tracked approximately the same. And until I added the Saccharomyces, the inoculated strain was the dominant strain. Does that answer your question?
Yep. Yeah. Thanks.
Okay.
Something else? Anybody else? Nobody.
Wow.
Ushka, from your side?
No, I would just maybe ask Megan, since she was using both targeted and untargeted metabolomic approach, if she can maybe describe a little bit describe about the weak and the strong point of each of the technique.
Oh, certainly. Well, starting with the targeted, again, the ability to quantitate can obviously give us, in a lot of ways, so much more information than otherwise. Getting at some of the questions that we've already had. Did the concentration of compounds that we see exceed the sensory threshold? There is going to be this lingering question of, just because we can see it chemically, doesn't necessarily mean it's going to really impact the final product. So being able to quantitate, I would say, was the strongest part of the targeted method. Unfortunately, we know that wine is a really, really complex matrix. 90 compounds, even though that sounds like a lot, is barely going to scratch the surface of what the overall profile of a wine is going to be. And it is unrealistic to think, at least right now, that we could quantitate over 1,000 different peaks. Now, that the untargeted ones, simply its weak point not being able to necessarily quantitate in the same way, does reveal some interesting insights. And I would say the strength there is, for example, that pyrrole that we found in the Lachantia in the Syrah. We wouldn't have seen that if we had just stuck to a targeted, very narrow analysis. And there were actually other things that I sort of skipped over for the sake of time that I detail in my published work in the dissertation chapters that we would not have seen that are worth investigating further. Another potential next step would be, okay, take the untargeted data, use it, and you can use the GCGC platform to do a targeted analysis, and you could go back and quantitate. If you run an authentic standard and then run your samples to get an idea of what concentrations you're seeing in your wine. That would be another really easy next step, which would really complement some of the failings of the untargeted method.
Okay, thank you. I don't have any further questions.
Thank you.
Okay, Megan, from my side, so from a data point of view, what do you think is our biggest challenges going forward? Because, I mean, technology will just increase the amount of data that we generate in future. Do you think bioinformatics is keeping up with the amount of data that we need to analyze using these technologies?
It is. Like bioinformatics, I would say, as a field, as a whole, is. But I would say one of the largest challenges I faced during this project was having to suddenly be something of an expert in not just GCGC and not just mass spec and not just biology, not just chemistry, not just winemaking, but then also adding this what is a very complex component to the mix of the data analysis and really understanding the current, I really should have had a sort of state of the art of bioinformatics tacked onto this study. And were it not for the really great support of my team, at Stellenbosch and at FEM to help me understand the data, I don't think we would have been able to answer some of these questions and to really be able to draw the conclusions that we did. And so I'd say while the field is keeping up, it is going to be very challenging for an individual student going forward to keep up. So it's really going to take a multidisciplinary and an interdisciplinary approach. It's going to take a team to really understand these more complex systems. And so I would say going forward, having a multidisciplinary team is going to
We've lost you.
Oh no. Come on. Can you hear me now?
I can hear you, yes.
Okay, that's fun. South Africa?
Yes, we're here.
Okay, good.
Are we trying to reconnect this establishment? I presume you're in charge of the panel now for the discussion. You can either do it now or tomorrow morning. Florian has just walked out, but I need it latest by 10 because I need to finish the report to hand in by 12 tomorrow. So if you can just drive that, please. But yeah, needs to.
So, sure, am I back?
Megan.
Hello?
Are you there?
Yes, hi. I don't think my video, for whatever reason, is going forward, but can you hear me?
Yes, we can hear you.
Okay.
So, I mean, obviously, I think we're sort of at the end. Is there anybody else that still has a question? If not, I mean, from my side, I would really like to thank you for your passion, enthusiasm, and self-driven attitude that you had towards your PhD. I mean, you're one of those students that I think kept your supervisors on their toes because by the time we thought of something, you've already normally done it or have at least investigated it. So it made life for us, I think, easy. And I know it was a huge challenge because the technologies and the data mining was things you had to learn. It was not if you had the necessary background. And I mean, I realized when you were here that there's only one way you do things, and that's just to dive into it and get it done. It doesn't matter who's in the way. If you need information or help, you will make sure that you get it to drive and get the information necessary. So I mean, from my side, I really want to say a big thank you. I know it's been challenging. Having 3 different places during your PhD and the final stage now in the States was not, I think, fair to say the most optimal normally, because finishing up a thesis and the writing, it's always much easier if you have the student on site. But I think you really handled it exceptionally well by, I mean, making effort on your side to ensure that you're up early or up late to ensure you catch us in office hours. So thanks for that. Megan has been in contact in Michigan now with the Brewing Institute, which wants to set up analytical laboratory, I think, to do metabolomics of beer. So good luck with that. I hope it pans out, that it's something that you want to do. And obviously switching from wine to beer, it still stays within the yeast community. It's just a different matrix and a different taste. So I think the skills that you've built up in your PhD will really suit what they're looking for. So yeah, the last is obviously we will still be in touch. Your last point was there's still a paper outstanding, which obviously your supervisors will drive again now. So yeah, from my side, I mean, I want to thank you for taking your PhD into your own hands and making sure that you were always on top of it. So that's from my side. I don't know, Benoit and Ushka, if you want to add something that might embarrass Megan. From a character point of view, Ushka, obviously she spent more time at FEM. Maybe there's a story or a joke that we're not aware of here in South Africa that you can tell about, Megan.
No, I think that Megan learned really a lot during her stage here. She came as a really young PhD, and she was growing a lot. She put a lot of energy in her PhD. She was following our suggestions, and I mean, it was nice following her because we were all growing during this PhD. So it was very nice experience. It was a little bit out of my field, and I learned a lot also during her PhD. So I'm very happy that she was my PhD student, and I think we had a nice experience.
Thank you.
So yeah, welcome back online. We have your face again. So yeah, from our side, obviously there's a panel that will sit now on your oral presentation that will give me feedback by tomorrow morning. Okay. And then I will let you know if it's all thumbs up, and we see you in March for your graduation, both you and Ushka, because Ushka's planning to fly out as well with Fulvio, as far as I understand.
We are working on it.
Okay. Well,
so
I will for sure be there. So I'm very much looking forward to seeing all of you and sharing a glass of wine.
Yes, so I know in South Africa there's a saying, as soon as the aeroplane has gone over, you can start drinking. I know it's early in the morning in the States, so, but I think you deserve it, and you can have a glass of wine now to celebrate this big achievement. So I will let you know tomorrow what the final is. But thanks a lot, and yeah, we will speak to you definitely tomorrow and for a while, or until we see you in South Africa at least, to finalize the thesis, which Karen needs by the 24th, and then finalizing the paper.
Okay. Well, thank you.
Enjoy your day, and we'll be in contact.
Okay. And thank you to everyone.